and then we'll get started with our presentation. We are the University of Arizona chapter of MEDLIFE organization. MEDLIFE stands for Medicine, Education, and Development for Low-Income Families Everywhere. This means that we serve low-income families here in Tucson and abroad, where our organization reaches. They are based out of Lima, Peru, and we have places all over South America and Africa where we go help out. Our mission is to help families achieve greater freedom from the constraints of poverty, empowering them to live healthier lives. This is a mission statement Gloria helped us come out with when she joined our team this spring. And this is essentially what our chapter is striving for specifically and what we are based off of. So moving forward, what is our purpose? Brady, Gloria, Max, I'll just let you come on in whenever you feel.
Sounds good. So basically, although we're a global organization, we like to help out locally. So in Tucson, we volunteer with a couple of clinics as well as projects that are designed to help low-income families and families in need. And basically, we believe that quality to healthcare is a basic human right. And because of this, we do everything in our power to help those in need.
Thank you, Brady. So we are the e-board. I am the president. I'll let you guys introduce yourself. Max, why don't you go ahead?
Oh, okay. I'm Max. I am the professional outreach chair. My job is essentially to help connect the student body of the U of A to the faculty and staff body. Yes, exactly.
Yep. So hi everyone, my name is Gloria. I am in charge of the professional development. So for this semester and the next semester, I'll be helping out with a couple of workshops on helping develop resume, cover letters, and practicing interviewing skills with other peers. So yeah, that's me.
I'm Brady. I'm co-president with Charlie. I'm a master's student here at the University of Arizona. And basically, I just want to work with everyone in this team to make sure everyone has whatever materials they need to apply to medical school and have the volunteer opportunities that they need.
Awesome. Thanks, guys. All right. So that's a little bit about us. And next, what we're going to do for you this year. So we'll go over some of our plans, some of the things that are kind of relevant to the entire year, and some plans that we have for professional development. So we'll go on with that. So obviously, there are some new standards of public health. We are fundamentally a public health club, so we're going to capitalize on that. So kind of our shout outs is that in general, follow CDC guidelines. We all are pretty familiar with those by now. So wear a mask, socially distance, order in if possible. Grubhub is awesome if you have a meal plan. Isolate as much as possible. So, partying is fun and all, but let's cool it until we can go back out and in full stride. And then what this means for the future of public health. So if anyone has something that they want to say about that, that'd be cool. We'll open it up for a few seconds. What do you think might be a future implication in the next year for these public health measures and how we go about handling contagious viruses?
So I think that the pandemic kind of gave us a good example of how we weren't too prepared to work together as a society to try and get through this whole situation. So I think that this is going to be a little bit of a learning experience for everybody, and hopefully it can be applied again in the future if something like this were to happen again.
Yeah, exactly. Thanks, Melissa. You're exactly right about that. Definitely emphasizing that there is a learning curve right now, but hopefully as we come out of it, we can definitely be better prepared for the next one. Because there's more to happen. This is just one of possibly millions of viruses. So, okay, how meetings will go. I'll let somebody else take this one on.
I can take this one. So because of the current COVID pandemic, everything's going to be online. I know there's a couple of transitions to online or to in-person stuff that's currently going on campus, but for the duration, it seems that we will be online unless we change. Basically, in a meeting, we'll share either interesting articles, helpful resources, or as today, we have Dr. Shwartz presenting on stuff and basically provide opportunities to talk to professors and engage with us, or basically, as Gloria does with professional development. Basically, we're a good resource for all things here.
Exactly. So we're going to focus on getting you ready for med school, getting you volunteer hours for really whatever graduate school you want to go to after this. Most of them require a lot of that, and we are trying to get those, so we want to offer those to you as well. So some of those being, so the Safe Walk is coming up. this coming week. And you can do this on your own, or we can come together as a group and decide that we're going to pitch in as a group effort and put it down as MedLife. We can still walk alone, but maybe we can do something like design our own masks or wear our own t-shirts. So that'd be cool if we could team up and do something like that. And we'll talk about it. We'll form a GroupMe page, or you can interact with us on social media and give us your insights. So that is coming up. That is the Southern Arizona AIDS Foundation. So we are walking for AIDS. And then the Safer Contact Tracing team, something I just joined. So that is through the U of A. They are contact tracing for the COVID-19 pandemic, calling students on campus and people in the local area. I think it is pretty close to campus, so it's going to be mostly students. But this does count as medically related volunteer hours, so that could be super helpful and also be a cool experience. And then, so usually we have our service learning trips, which go out several times a year. And in the last semester in March, I was supposed to go to Lima. And that kind of went out the window, obviously, with travel orders. And to mitigate that, what MedLife organization is doing is offering a virtual service learning trip experience. So in that, you're going to be able to interact with professionals and community members online. It will count for a total of 20 hours. So you'll be able to get a good understanding of global health. A lot of people there that have been working in Lima have worked there for several years now. We've had the pleasure of working with a few of them personally, and they really know what they're talking about on many levels. Again, medicine, education, development, and these people are involved in all three of those all the time. So they're really great people to talk to, really great people to learn from. So you'll network with them and create connections with other MedLife chapters in the process. So usually we'll team up with a few other schools and do this together. So this one is only $249. And part of that, or all of that actually does go to donating for the families there. A lot of people in Lima, Peru specifically, don't have official employment. So a lot of these people aren't getting any type of relief, and a lot of them are struggling to get food. So what MedLife has been doing over there is providing a lot of families with meals, and that's where that money will go as part of your participation. So where to find more information about that? We'll watch a little video here, but then if Gloria, you can click these links and post them in the chat, we'll give you the information on how to register and how to learn some more information about it. And this video is pretty quick. It's just a couple minutes, so bear with me here.
Hey MedLifers, my name is Tim Anson, and I want to invite you to join us in our new virtual service learning trip, Med Anywhere. All right, let's go. In Peru, we've been on government-enforced lockdown for over 180 days. But that hasn't stopped MedLife from responding to the crisis that COVID-19 has created. The virtual SLT will give you an inside look at our work, the realities of the virus in Peru, and more. For a week, I will guide you through different communities in Lima, food deliveries, and soup kitchens. I will introduce you to community members so they can share their stories, and to MedLife staff who can show you how we do our emergency response and patient follow-up during the pandemic. I will also host discussions and engaging workshops to give you a hands-on learning experience from your home or your clinic. We are currently in the community of Union Santa Fe. This is one of the communities that we talk about the most because it's where we have the majority of our projects. As you can see behind me, for those that have been part of the World Unified Project, that's where we stand with that. Here is the Mini Med Center. Now, this is something that we have really overhauled since the start of the pandemic, or since mid-March. And what we're able to do here is provide food to the community. And now we're going to go inside the Med Center to Beta. And so let's see if that goes down.
Hello everyone, I am Roberto Paredes from MedLife. I'm here in the third floor of the Med Center, in the area that is the bodega, and also the area you can see behind me is the internet for the community.
This trip is not only a great virtual travel and learning experience, we will also gain 20 certified service hours with your participation. And the best part about participating in a virtual service learning trip, you will directly contribute to our goal of serving 1 million meals in communities facing food insecurity due to COVID-19. Your virtual volunteering will also support our year-round medical, education, and development work. What are you waiting for? Signups are now open on our website. I hope to see you soon.
Okay, so hope that cleared up any questions you might have. These spots are super limited. I think they're only doing 60 at a time, and that might be only for the semester depending on how it goes. They are kind of worried about the Wi-Fi connection and other certain specific implications of being over there. So do sign up quickly, and then I'll pass it on to Gloria.
Yeah, so a couple of goals. We're offering all these opportunities, but as the professional development chair, I want to host workshops to be able to show you how to best represent yourself in a professional manner. So in a resume, it's a one-page representation of your most important accomplishments. And depending on what position you're applying to, I can also help you tailor your resume to fit the position. In a cover letter, it emphasizes 3 skills that you'll discuss by talking about stories from that employment. And it's kind of your way to prove that you're able to exercise these skills in a professional manner, such as a job. And then I also want to practice interviewing skills. And so I know there's a lot of mini mock interview skills that we can do. And so especially in pre-health fields, this is so important to be able to talk about your career and what your strengths and weaknesses are and prepare an elevator speech. So I'm really excited to like set this goal up for our club.
Awesome. Thank you, Gloria. And then, so here are some of our other speakers. I'm familiar with Dr. Kennedy. He's awesome, so I can't wait for that. And then, of course, I'm excited to meet these other professionals that Max has set up for us. But Max, I'll let you take it away and introduce.
Yeah. So the speakers that we'll have throughout the semester, they're either going to be involved in the research field or the medical field. And so we actually have the privilege today of having Dr. Shwartz here today. And he's going to talk to us about his research project that's centered around Ewing sarcoma. Yeah. So without further ado, here's Dr. Shwartz for you.
Excellent. You guys hear me?
Yeah.
I'll share my screen here. All right. And you see a PowerPoint, right?
Yeah. Yeah, we do.
Good. Okay. Well, I'm excited to be here today. I was very glad that Max invited me and to get to speak to you guys about our research looking at a type of cancer called Ewing sarcoma. It's a pediatric cancer. So I'll start with what is Ewing sarcoma. As I mentioned, it's a pediatric bone cancer. It is driven by a fusion protein, an oncoprotein. I'll explain what that is. It's the second most common bone cancer in children. And one of the unusual things about it is that it often occurs in late adolescence, so like between ages of 10 and 20, so teenage years. It occurs more often in males than females. It tends to target the long bones, possibly just because of their mass. So 25% of the cases are metastatic at diagnosis. So the prognosis is not terrible as far as cancers go. If it's your family member, though, 75 to 80% survival is not encouraging. But those cases that are metastatic, the survival rate is very low, 30% or lower. And so this cancer hasn't seen an improvement in outcomes in over 30 years. So there's limited options for treatment. They respond well to chemotherapy and radiation, and for these types of bone cancers, they typically also just go right to amputation. In addition, because these are aggressive cancers, as you can see, the fact that so many are metastatic at the point that they're diagnosed, I think alludes to the fact that this is a very aggressive cancer. This cancer is driven by proteins that are associated with this FET family of proteins. So what I'll talk to you today about, we'll focus on this one called FUS. It's named for fused in sarcoma. FET is FUS EWSR1, TAF15. EWSR1 is Ewing sarcoma related. And TAF15, it was discovered as a transcription-associated factor, so that's what that name is for. We'll focus on the first two, FUS and EWSR1. What's interesting about this is half of all known translocation events, so that's where a break occurs in between two chromosomes, the sister chromatids, and there's or one of the sister chromatids, and there's a fusion of one chromosome to another chromosome. And this can result in a fusion protein. So half of these events that are known to occur to drive sarcomas involve one of these three proteins. The oncoprotein that is produced by this fusion is a very powerful oncoprotein, very tumorigenic. So looking more closely at this, about 85% of Ewing sarcoma is a fusion between a protein, that protein EWSR1, and a developmental transcription factor called FLY1. About the remaining 15% that's where you'd find a lot of the FUS fusions that drive Ewing sarcoma, as well as some alternative EWSR1 fusion events. EWSR1 and the rest of the FET proteins, they're ubiquitously expressed. They're expressed in all of your tissues. They're conserved throughout vertebrates, and a FET protein is found actually all the way through multicellular organisms. And that is a good point to highlight how important it is to biology, so that every organism, as soon as they became multicellular, no one's figured out how to survive and how to have their cells thrive without a FET protein like FUS. And since vertebrates arose, none have figured out how to live with less than 3, which I think also highlights to us, despite the fact that they can cause similar tumors, that they must have some kind of independent role that we're just now learning about, since the cells always have to have 3 of them. So that's EWSR1, and then the protein FLY1 is, as I mentioned, a developmental gene. So after development's done, this gene is usually turned off. So it's not supposed to be turned on after development is over. And that's exactly true, that this gene lives on chromosome 11, EWSR1 lives on chromosome 22. What you're going to see in Ewing sarcoma, this fusion event is going to take part of the EWSR1 protein, the N-terminal part, and fuse it to the DNA binding domain of the FLY1 gene. And so this has a functional significance. EWSR1 and the rest of the FET proteins, the C-terminal bits are the RNA binding bits. These proteins' activity is normally regulated by RNA. They regulate transcription. They find where transcription is happening by finding where RNA is being made. Swapping out those RNA binding domains for a DNA binding domain essentially short circuits the system. And so now this fusion protein is going to be driven to wherever this DNA binding domain wants to bind. And this fusion protein is able to dominate transcription. It can overrule all the cell's normal control pathways for gene expression and drive it towards tumorigenesis. So this is what the translocation looks like. So there's an actual break in the chromosome. It's in the middle of the gene that codes for EWSR1. And that part of chromosome 22 gets mistakenly fused to chromosome 11 when that break is being repaired. And part of chromosome 11 gets fused to chromosome 22. Since EWSR1 is the gene that's supposed to be expressed, the fusion that starts with EWS and ends with the FLY1 domain now gets turned on, even though FLY1's normally not expressed. And this can both turn transcription on and turn transcription off. So why this relatively rare cancer is so important is because usually cancers are driven by a panel of different mutations. You can think of it like a bingo board. Unfortunately, though, bingo means cancer. So you don't want to get bingo. These are unusual. These proteins are so powerful. This is a one-shot, one-kill cancer. This is the only mutation you need. These tumors are sensitive to DNA damage, so they very rarely have other mutations. As few as 20% of the tumors have secondary mutations have been identified, other than EWS FLY1. Why that's important is that this seems to be representative, and that's the hypothesis, that this is showing us a least path to tumorigenesis. So we can follow the molecules from the initial genetic insult all the way through the genes that you need to control in order to result in tumor formation. Not only that, a very aggressive tumor that likes to metastasize quickly. So it's because this is a simplified system that we can easily track mechanism to result. The thought is that studying this system will teach us a lot that will immediately apply to other tumors, other cancers, other sarcomas. So what do the FET proteins do? What I'll tell you about is that these proteins are RNA binding and they regulate transcription, and they do it through this weird process of oligomerization. So the proteins binding to each other. I saw Gloria mention that she likes artwork, and I did not share my slides, so I use this as my example. So my lab studies biochemistry. I speak a lot to, just on Monday, I was speaking to a group of Ewing sarcoma clinicians that are actually treating patients. They're often looking for genes that cause disease. The difference between genetics and proteins is genetics is a very abstract concept. It's a gene itself is an abstract concept as a location in the genome, whereas a protein to us biochemists is a very physical machine. It has a defined chemistry, like a truck. And so there's a very real relationship between structure and mechanics and function. So why that's important is when you break a protein, there's only so many things a protein can do. Just like when you break a truck, it's still a broken truck. There's no way you could break a truck and have it turn into a tree. You can strike this truck with a Hellfire missile. It is still a very broken truck. So understanding what the original proteins were supposed to do limits and guides you to understanding what the disease proteins are doing. Whereas to geneticists, they break the world into gain of function and loss of function, as again, these are abstract concepts to understand mechanisms of disease. To us biochemists, since we understand the mechanics of the original proteins, the disease state is usually just an exaggeration or a misdirection of what the proteins were originally designed to do. So what these proteins do is they're part of a family of RNA binding proteins. So when your genes are expressed, you'll recall DNA is copied into RNA, and all of your protein coding genes are transcribed by this polymerase you'll see several times throughout the talk, RNA polymerase 2. I'll call it RNA pol 2 for short. The RNA, while the RNA is being made, you learned about processes in your classes like splicing. You might remember that RNA can have a poly A tail in higher eukaryotes and in mammals. That's just a string of A's on the end and a cap. Those are important for controlling the fate of where this RNA goes and what protein it codes for. All of that processing is done while transcription is occurring. And so the proteins, like the FET proteins and the other H and RNP proteins, are loaded onto the RNA while the polymerase is moving. This is an incredibly complex process that's not well understood because you require hundreds and hundreds of proteins to find that RNA while that polymerase is moving at 4,000 base pairs a minute. And they have to do their job like splicing and all of that while the polymerase is moving. How that is organized is a big question in science today. We believe that the FET proteins are part of the answer to how you organize that process. So the FET proteins, they basically broken into two parts. This is the part that gets fused onto these cancer-causing oncogenes. They have this amino acid motif, like a serine and a glycine with a tyrosine in the middle, followed by another serine and glycine. It's repeated over and over. And these also are simple repeats that it uses to bind RNA. Together, these domains allow the protein to undergo what we've now come to call phase separation. This has been a discovery just within the last 5 years. It's a long-standing question, though, because obviously you've probably seen in pictures in your textbooks of cells, an example of phase separation of the cell is like nucleoli. That's where your ribosomes are synthesized in the nucleus. This is an organelle in the cell that, unlike the nucleus, there's no lipid membrane around it. So the cell is bounded by a lipid membrane. Mitochondria, Golgi, all have lipid membranes around them. These organelles, it's just spontaneously proteins and nucleic acids come together and concentrate themselves in the cell. Or spontaneous organization, that's what you're looking at. Well, the big discovery when we started learning how to control and purify and study in the test tube, the FUS and EWSR1, we found that these proteins in the test tube can do this process by themselves. So it's the first time in the test tube we could watch this organization occur outside of the cell and study what the chemistry is that's driving that. Interestingly, these also recapitulate a lot of the observations in the cell. These condensates, we've come to call them actually, that term's largely been agreed on just in 2020. This is a fast evolving field. You can see they have kind of liquid-like properties. You see these droplets, we took in our lab these pictures just August. The proteins form these liquid-like, it's like oil in water, you could think of, like drops of oil mixed in water. They can fuse together. Here you've caught in this picture 2 droplets fusing together. These have not quite fused together. Here you can see them like pinching apart. Again, it looks like dew on the hood of your car. These things kind of self-assembling. So in the cell, there's a lot of bodies that are found, stress granules, P bodies, I've mentioned nucleoli, Cajal bodies, paraspeckles. They're all thought to be formed by this process. However, when I started my lab, there was a problem that there was no body that was associated with transcription. And I had worked understanding how this process during my postdoctoral work before I came to U of A, how this process here is associated with transcription in the test tube. But there was nothing like this that we knew of in the cell. So is there one of those condensates in the cell? The answer is yes. Now there's actually a number of labs working on it. So a number of people have been able to identify these within the last 2 years. The papers have started coming out. So the initial discovery that I worked on during my postdoc was that the FUS protein protein binds RNA and undergoes this assembly process. So this is the process that we refer to as oligomerization. So the proteins are binding one to another. The RNA, this term seed we took it from like the amyloid formation you might be familiar with in Alzheimer's, Parkinson's disease. Biochemically, there's some similarities to the way these fibers form and the way amyloids also form. However, though, unlike amyloids, this is the normal function of the protein. So when the protein is in the state I showed you here, and that was the discovery we made in the test tube, that's the active form of the protein that can do its normal function. So unlike amyloids, this is the protein doing what it's supposed to do. As we came to more recently understand more about what these oligomers form and the forces that drive them in the cell, we've come to realize that fibers that we can recreate in the test tube probably don't accurately reflect what's in the cell. Instead, in the cell, they form much looser interactions, and so they can adopt these liquid-like droplet states. So in 2018, my lab published their study of isolating so we could actually physically look at one of these granules from the cell. They're so small that to get compelling evidence of their existence using microscopy is very difficult. And in fact, as people develop new microscopy and new techniques, it's kind of like, you know, the ultimate test that your microscopy is cool is to try to see the polymerase in action in the cell. So that's a test that a lot of people who are developing new microscopes put their microscope through. But we decided to go through and stabilize these by using chemical cross-linking so we could take these particles out of the cell and look at them with, this is electron microscopy outside of the cell. So RNA pull 2, this is a structure of RNA pull 2 with the DNA inside of it. It's 25 nanometers across its longest axis. When we pulled out these particles that are associated with pull 2, there's a pull 2 holoenzyme. This is the granule or the droplet or the condensate, if you will, that's associated with RNA pull 2. You could imagine you can fit a lot of other proteins in there, which is a good thing because this here gives us an idea to conceptualize how this organization happens in the cell. We know from our work in vitro that these are very porous. In fact, you could think of them like a molecular sponge that you can tack on and have many other factors that are required for transcription to be prearranged and held close to the gene that's being transcribed.
Yeah, Dr. Shwartz, I don't mean to interrupt you. I actually have to head off to a class, but it was nice seeing everybody. And thank you once again, Dr. Shwartz.
Great, thanks. Well, so these factors here, that's the way we originally thought of these as being a way to bring these together onto the gene. And so we thought of that's what's going on inside of this granule. So the hypothesis was in Ewing sarcoma that this fusion protein is hijacking this process, taking these normal granules and taking them to where the fusion protein wants them to go. And that's how it's so powerful to dominate, because it's taking all the transcription machinery that it needs with it. So as I said, these are like a molecular sponge. So they have these oligomerization domains. This is the RNA binding FET protein. This is the fusion protein. This is short-circuiting the system. The fusion binds the DNA. It binds GGAA repeats and can bring together through that oligomerization domain, this was our hypothesis, the FET proteins and form a granule. So to test that hypothesis, which turns out we believe is true, the first thing we did was we looked in Ewing sarcoma and asked if we removed either the EWSR1 FET protein or the fusion protein, if it's that interaction is functionally relevant, we should see similar effects for many genes when we take away one or the other. So this is just testing that we got them taken out. We took out one, the fusion protein, the other, the EWSR1, and here we reduced the levels of both proteins using a process called RNA interference or siRNAs. Then we used an experiment called RNA-seq. It's a way of measuring gene expression for all genes in the cell all at once in one single experiment. Obviously requires a lot of computational development or analysis of this types of data. So what I'm showing you here is this is from our study where we knocked down either the EWSR1 or the FLY1 or both. This is a typical set of genes that's called the transcriptional, if you will, fingerprint of Ewing sarcoma. And so comparing this to all these other studies of other Ewing that are also done on Ewing sarcoma, you see the same panel of genes are consistently turned on when you lose the fusion protein. And what we had found is that many of those same genes are also turned on when you lose the wild type protein, EWSR1, suggesting that they're working together. And that's also indicated this way. If you plot them, the changes for one knockdown on one axis change for the other on the other axis. In green, these are the ones that are going up or down in the same direction, whether you knock down either. So there's hundreds of those genes. The next function we tested is tumorigenic, or cells that have acquired tumorigenesis. They're able to grow on non-adherent surfaces, and we can mimic that in vitro by taking cells. These are cells from Ewing sarcoma patient. We can put them on, instead of growing them on like a plastic dish, we grow them on agarose. Non-transformed cells can't grow on agarose because they need something to adhere to. Cancer cells, and particularly the more metastatic they are, have no problems. When you remove the fusion protein, you find that this activity is compromised. The cells can no longer grow in a non-adherent manner. When you move the wild type protein, it's, with this knockdown, it's even worse. So again, this is a functional relationship that looks the same. If you take another non-Ewing sarcoma cell line that doesn't have the fusion protein and you remove the wild type protein, EWSR1, you don't see this compromised growth, this compromise in this transformative ability. So that losing the ability for these cells to grow in a non-adherent way is not a general feature of losing that wild type protein. To make sure we understood what was going on, we took these cells that are not Ewing sarcoma cells, we put in it the gene that drives Ewing sarcoma. And all of a sudden that recapitulated. Now when we lose the wild type protein, now the cells can no longer grow on a non-adherent surface. So they've lost their tumorigenic capacity by losing this wild type protein. Again, highly suggestive that the wild type protein and the fusion protein are working together. We did a bunch of studies to study interactions, and we used our cross-linking technique because these granules or condensates in the cell are so weakly held together that when you break the cell open, they immediately fall apart. Whereas like normal protein complexes like RNA-Pol2, those are strong interactions, and so you can purify this, solve a structure using crystallography. These are difficult to purify. So we developed this cross-linking technique to get them out of the cell. Then we're going to be seeing some pull-down experiments where we pull down one protein that's been cross-linked and we look if the other protein has been cross-linked. So this is showing evidence that they could be together inside a granule body or a condensate. First, I'll show you just the wild type protein in POL2. This was an expected result. And so this is to confirm by cross-linking when in a non-Ewing cell, you can see this is the control. There's pulling down the polymerase. It's an antibody that recognizes a phosphorylated form of the polymerase. Pull down the polymerase, you get the wild type EWSR1. Pull down EWSR1, you get the polymerase. In the Ewing sarcoma, same thing. And in fact, in Ewing sarcoma, these experiments are rather hard to do, but when you use the cross-linking, it works much better. Next, we looked at the three together. What about the wild type, the polymerase, and the fusion protein? Here in this experiment is one of the ones where we put the fusion protein in a non-Ewing cell. When we do that, you can see we pull down EWSR1, we get strong signals for the fusion protein, pull down the polymerase, also get strong signals for the fusion protein. Then over here, we can also see the same thing in the Ewing sarcoma. And these experiments, as I mentioned, these are pulling down the fusion protein in Ewing sarcoma is pretty difficult to do because the interactions are so weak. But with the cross-linking, we're able to recover these complexes. Now is the big one, can we see a granule formed with the fusion protein? So we used our cross-linking, we pulled out the fusion protein from cells, and here you see these round bodies formed. This is looking at the different sizes. Interestingly, the fusion protein tends to associate with somewhat relatively small, much bigger than a normal protein. I told you POL2 is like 25 nanometers. The average protein by itself is like 5 nanometers in diameter. So POL2 is like one of the biggest machines in the cell. But these can be quite large, up to like 80, 85 nanometers in diameter. So what does this mean? Well, there's a lot of things we could have found in the cell. Over here, I'm showing you in the same experiment as a positive control. We also pulled down RNA-Pol2 for a direct comparison. You can see here all the different particles that we quantified their diameter. Over the years of studying these particles that these types of proteins form in the test tube, we've seen a lot of different structures formed. We've seen fibers formed. We've seen fibrils formed. They form the normal protein forms in an RNA-dependent manner, which is something that we've shown to occur in the cell. So we could have seen of the fusion protein, we could have seen fibers like this. We could have seen fibrils. This is another type of fiber structure that was made a fusion of Fly1 with a long strand of DNA repeats. And this is kind of like a ball of string falling on itself. And this is a type of structure we refer to as a hydrogel. It's a stiff version of these liquid-like properties. But it looks like what is actually formed in the cell are these liquid-like granules. So this experiment was also very informative about the physical properties that these proteins make. So with that, where we're going in the future is we think that this new understanding of how transcription works, as well as the current frontline therapy is chemotherapy, DNA damaging chemotherapies and radiation. Like one of the projects we're doing in my lab is combining transcription inhibitors and DNA damaging chemotherapies, because we know these proteins, we know the mechanism that they're playing a dual role in both processes in the cell. And we've gotten really good results that we can get greater lethal effect on the tumors and not non-Ewing sarcoma cells by doing these types of combination therapies. And it stems from us understanding what the mechanism is, how these proteins work. Well, with that, actually, as Max was referring to, this was a picture of our lab on the top of Blackett's Ridge. The work I showed you today, the work in Ewing sarcoma was done by undergraduate, Lucas, and the CEO was the graduate student. The work with purifying, the initial work to learn how to purify these granules out of cells was contributed. Actually, there was a challenging experiment, so a lot of people worked on it. Val, in what I refer to as Val's army. So Rachel worked on it, Matt worked on it, Vivian worked on it, Karis and Daniel have worked on it. So there's a collaboration across the lab. And with that, I'm happy to take questions if we still have time.
Wow, Dr. Shwartz, that's some amazing research. Like you said, there hasn't been a whole lot of progression with Ewing sarcoma in over 30 years, I believe you said.
Yes, yes.
Very cool. And then I also work with Daniel Wayland, so neat to see that.
I'm sorry, what's that?
Oh, I said I also work with Daniel Wayland. I'm in a club with him as well, so it's neat that he's on your team.
Nice, nice. Yes, yes. He's been great. In fact, the next part of this story that we're preparing the manuscript now was a project that Daniel was very much involved with in getting those protocols. We've been able to now not just recreate the individual components like the interactions and the condensates I showed you. We've been able to, and Daniel helped with that, put them all together and actually do transcription in the test tube. So we can watch these form as transcription's occurring.
Oh my gosh, that's incredible. I'll have to ask about that.
Can I ask a question, Dr. Shwartz?
Yes.
So I know there's been like a little bit of a debate of what polymerase factories are. Would you mind giving some insight into how the liquid-liquid condensates correlate or like either support or just repeat that theory?
Great question. So, you know, it was all the way back to like around the turn of the millennium, like the start of the 21st century, people could do the math. And even if you modeled in the known features, the tricks that the polymerase plays to find a gene in time, like when you have an immune response, COVID-19, genes are turned on within hours. But if you do the math, the number of molecules in the nucleus, not to mention the hundreds of those that have to come together to find the gene, to turn a gene on, it doesn't work out. By random chance, they would never find the DNA in time. It'd take days for you to mount a response like for your immune system. So the idea was there must be some way to pre-organize, but our understanding of molecular scaffolds was based on like microtubules, actin. They can be pretty dynamic. You can watch cells moving across a surface. And those are, you know, these structural proteins like actin and tubulin moving. But the polymerase is moving so fast. There was a real problem and a lot of doubts about that, is what kind of rigid structure like this could accommodate this polymerase that's moving over enormous spans of the DNA. Mammalian genes are typically, you know, tens of thousands of base pairs long in the genome. When we found these liquid-like particles, all of a sudden we were struck by, this is something that's fluid enough and dynamic enough that it might be able to provide a scaffold that can accommodate all the moving pieces, like the moving polymerase. I did put poor Rachel on the first study. I asked her to see if she could test how fast it could polymerize and assemble in the test tube, and to the sizes that we were able to pull out of the cell. She went as fast as she could pipette, and she could monitor them forming within like sub-minute time regimes, like down to 30 seconds. That's getting in the ballpark of how fast it needs to move in order to do the test tube. But the limit there, like I said, is just how fast can you pipette. So I think we're going to actually have to develop new techniques to see this happen at the rate that the polymerase is going. So that was the important, the fascinating discovery, is that this is a process that's so dynamic. It could solve the problem of organization, but accommodate all the moving pieces when you're transcribing a gene.
Thank you.
All right, last call for questions. Well, thank you so much for coming today, Dr. Schwartz. We really appreciate it. Sounds like some really amazing, groundbreaking research. So look forward to seeing it progress. Thank you again for coming today.
Thank you very much for having me. It's a real treat. You guys have made my week. I was really excited to see the work you're doing and the plans you have for your volunteer work.
Well, thank you. We appreciate that.
All right.
All right. Thank you so much. And for anyone else, if you're interested in joining our board, there's the link to sign up and become an eBoard member. We'd love to have you. We're doing a lot, and it's a lot between 4 people. We feel like we could do more. So join the team. All right. Thanks, everyone. Have a good week.